Myself Arvind Kumar, Assistant Professor of Botany, LND College, Motihari, and in this video lecture, I am going to discuss about the structure as well as the function of the Polytene chromosomes, which, as all of you know, must be knowing that it is a type of giant chromosome, which are very large in size. So first of all, we will know about what is introduction. So as all of you know that polytene chromosomes are giant chromosomes which are very common in some dipteran flies such as Drosophila etc. They were first observed in the year 1881 by E.G. Balbiani in Chironomus and were studied by Koschelt in the year 1884 and Corney in the year 1884. Heidenbauer in the year 1933 studied these giant chromosomes in Vibrio hortulanus larvae, while Painter described them in salivary glands of Drosophila. These chromosomes are generally somatically paired, so they never are found alone, they are always paired. And the number of giant chromosomes in salivary gland cell appear to be half that in normal somatic cell. So giant chromosomes are generally half in number in comparison to the normal somatic cell, but they are much larger in size. Now these giant chromosomes called polytene chromosomes, they occur in rectal epithelium as well as Malpighian tubules of Drosophila. They are much larger than the normal chromosomes, reaching up to a length of 200 micrometer, and they are even visible under a compound microscope. They become so much larger. And since this chromosome actually consists of many strands, they have got many strands. That is why they are called as polytene chromosomes. The polytene chromosomes are oversized chromosomes. They are much bigger than the normal chromosome, and they have developed from the standard chromosome. A specialized cell in certain organisms, they undergo repeated rounds of DNA. These specialized cells undergo repeated rounds of DNA replication without cell division. That increases their cell volume, forming a polytene chromosome having multiple strands, and the condition is described as polytene. Now I will discuss about the morphology of the polytene chromosome. The polytene chromosome, as I have already told you, these are much larger in size due to presence of many longitudinal strands, and these strands are known as chromonymata. The many strands of giant chromosome is due to repeated division of chromosome. These multiple strands are formed due to repeated division of chromosome without undergoing cytoplasmic division. And this type of division is called endomitosis. The polytene chromosome also contains two types of bands. These are banded structure. They contain two types of bands. The dark band, which is generally 80%, which contains 80% DNA, and the interband, which contains around 15% DNA. Here you can see the structure of a polytene chromosome of Sesidomya serotini. Here you can see this is the nucleolus and this is the interband and band, the banded structure of the polytene chromosome. Here you can see at the end there is a heterochromatin structure. In this diagram, you can see this is the normal entire chromosome, the whole chromosome of the organism. And here you can see the chromosome which is showing the enlarged part, that is the puffs which are formed and knobs or bands which are formed in the polytene chromosomes. As I told you, the polytene chromosome contain dark bands and the light bands, and these form important part of the polytene chromosome structure. The dark bands are darkly stained, as the name suggests, and the interbands are lightly stained with nuclear stains. The dark bands, they generally contain more DNA and less RNA. As I have already told you, they contain almost 85% DNA. The bands of the polytene chromosomes become enlarged at certain times to form a swelling known as puffs. The swelling of the bands at certain points is called puffs, and the formation of these puffs is known as puffing. It has been seen that during the larval development of the Drosophila, a specific areas on polytene chromosome they become uncoiled, forming localized region called puffs. The puffs represent regions of active RNA synthesis, also known as transcription. So these puffs, which become uncoiled and they become very large in size, these are actually sites of transcription or RNA synthesis. In the puff, individual fibers remain continuous across the puff and they become extended as short lateral loops, according to Barr 1954. So these puffs remain continuous, but sometimes they become extended as short lateral loops. DNA is concentrated almost entirely in the bands, and protein and RNA is also found in puffs. Puffing is generally, it happens due to the uncoiling of chromosome fibers, which are usually closely folded or coiled in the dense band region. These fibers then project in the form of loops. Now, as I have already told you about the puffs, there are certain other structures which are formed in the polytene chromosomes, and these structures are known as Balbiani rings. Now here we will understand as to what is a Balbiani ring. So as I told you, the puffs are formed during their initial stages of development. These bands or the interbands of chromosome, they exhibit swelling or puffs. Their appearance depends on the stage of larval development. So larval development stage decides the appearance of puffs. It is probably that the metabolic activities required for the formation of puffs are related to the secretory function of salivary gland. That means the salivary gland, its metabolic activity increases when more saliva is required. So what happens when more saliva is required, its metabolic activity increases, and due to this more puffs are formed, more RNA are formed. The formation of this is controlled by certain specific genes, and the puffs are related with the active synthesis of RNA and proteins. This chromosomal RNA differs from the nucleolar or the cytoplasmic RNA. The chromosomal RNA, which have separated in the form of puffs, these are very different from the nucleolar RNA or the cytoplasmic RNA. The RNA of the puffs is also not similar. It differs from each other in chemical composition. Chemical composition also varies. And some regions of the puffs, that is larger puffs, show larger puffing, and these regions are called Balbiani ring. So what is Balbiani ring? It is the puffs which have become larger than the normal puffs. So these puffs, which are very large in size, are called Balbiani rings. And what is the function of these rings? These rings are formed by the lateral stretching of the loops caused by chromonimata. These loops of chromonimata make up the Balbiani rings and give the chromosome a fuzzy outlook. Due to presence of Balbiani ring, the chromosome gets a fuzzy outlook. And the Balbiani rings are rich in DNA as well as messenger RNA, and the formation and function of the Balbiani rings are similar to the puffs. Here you can see the structure of a Balbiani ring. Here you can see the dark band. This is the central part of the chromosome. This is the interband, and this is the chromonimata, and the whole structure around this structure is called Balbiani ring. And in region of puffs, the chromonimata uncoil and open out to form many loops. In the region of puffs, what happens is the chromonimata uncoil and open to form many loops. The puffs indicate the sites of active genes when messenger RNA synthesis takes place. So what is a puff? It is the site of active genes when messenger RNA synthesis is taking place. The intensely stained chromosomal segments correspond to high degree of packing and are generally inactive. So intensely stained chromosomal segment, I am repeating this, intensely stained chromosomal segments correspond to high degree of packing and are generally inactive, and these regions are called heterochromatin. So heterochromatin region is inactive region, and they have got high degree of packing. Now there is another region which is of less degree of packing or less lightly packed, and these regions are also stainless. That means they are more darkly stained, less darkly stained than the heterochromatin. And these active regions are called euchromatin, which are less tightly packed and less stained. In Drosophila, it has been found that 5,000 bands have been found in 4 chromosomes of salivary gland cells. And chromosomes in the bands are at right angles to the long axis of chromosome. So chromosome is, that is polytene chromosomes, are situated at right angles. And that means at the 90-degree angles to the long axis of chromosome. And bands are having high DNA content and absorb ultraviolet light. And these bands contain 5 long arms and 1 short arm radiating from a central point called chromocenter, found by the fusion of all the 8 chromosomes found in the cell. The short arm radiating from the chromocenter represent the 4th arm. One of the long chromosome is due to the X chromosome, while the remaining 4 arms represent the arms of the 2nd and 3rd. So here you can see all the arms. This is the longest arm, the X chromosome, and this is the center. At the center you will find there is a chromocenter which is highly colored, and these are the different arms, chromosome 2, chromosome 3, and as you can see, since these are found in pairs, so you can see there are left arm and right arm, left arm and right arm. So these you can see, and some of the chromosomes are not paired. So this is the structure of the polytene chromosome of Drosophila. Now, what about the function? Why these polytene chromosomes are formed in Drosophila? They must have certain functions. So, according to various scientists, in addition to increasing the volume of the cell, because these chromosomes are very large in size, when they are formed, they increase the volume of cells nuclei and also causes cell expansion. These polytene cells may also have metabolic advantage. They don't only are formed only for increasing the size of the cell. They have got certain metabolic advantages, because since multiple copies of genes are formed, at increases. And the second function in case of Drosophila melanogaster is that the chromosomes of the larval salivary gland, they undergo many rounds of endo-reduplication, to produce a large amount of glue before pupation. Since this endo-reduplication produces large amount of puffs or Balbiani ring, and these Balbiani rings or puffs, they help in formation of more RNA and more protein, which are required for the glue formation, which are just the necessary requirement before pupa formation in case of Drosophila. This glue is required for pupa formation. So what happens? The polytene chromosome help in uh, the glue formation in Drosophila, and this is one of the major function of the polytene chromosomes. So this is the end of my lecture, and I think I have discussed all the matter. Thank you very much.